All right, so this video is going to cover questions 6 through 10 of the life science practice test. So let's look at question 6 here. Individuals in many parts of the world are typically immunized against tuberculosis, called TB. But immunization against TB in the United States is rare. Which two, so we need to pick two answers here. Which two of the following are the most likely reasons why TB immunization in the United States is rare? So again, two answers. So A, individuals worry about possible unintended effects of vaccines. B, the incidence of TB is relatively low in the United States. C, a TB skin test cannot be used to diagnose disease in an immunized individual because the results are always positive. And D, the vaccine is prohibitively expensive in the United States. So again, let's go through and look at the wrong answers first. So in this case, there's going to be two wrong, two right. So A is one of the incorrect answers because while it is true in our current world that a lot of people are more hesitant about vaccines than before, there's no science to suggest that there are significant side effects from the vaccines. Before a vaccine is approved for the public, the risks of the vaccine must be significantly less than the risks of the disease itself. So again, in a kind of post-COVID world, there are some worries about possible unintended side effects, but that's not why we don't immunize in the United States. And then the other correct answer is D. The vaccine is prohibitively expensive in the United States. This is incorrect because other countries less well off than the United States are giving that vaccine. So obviously, cost is not an issue if countries that have less money than we do are offering this vaccine. So A and D, not correct. So the correct answers here are going to be both B and C. So let's start with B. B is correct because TB is very rare in the United States. So since it is so rare, there's not a serious need to vaccinate. Other countries may need to vaccinate against it more because it's more common than it is here. C is also correct because how a vaccine works is that it shows your body a small part of the virus that's not active. So it can't hurt you or get you sick, but it presents that small part of the virus to your body. And when it shows it to your body is able to produce antibodies against it. And therefore, it allows your body to be ready to fight it if it does come in contact with the actual virus. So if you get a TB skin test, the way that works is they inject a little bit of the virus, again, not an active virus, so it can't hurt you, but they inject a little bit and they see how your body responds. So if you've had the vaccine, your body is going to react because you have the antibodies in the same way that your body is going to react if you have or had TB in the past. So not giving a vaccine is the only way that we can test if your body is going to react to it. Because again, once your body is going to react, if it's either you've had TB, you currently have TB, or you've had the vaccine. So because it's so rare for us in the United States, the idea of giving that vaccine and then not being able to test is also a problem then. All right, so let's look at question 7 now. A virus infects only some types of cells in certain animal species. Which of the following is the greatest importance in determining the cell types that are infected? So A, the structure of a ligand on the surface of a virus and a receptor on the surface of the cell. B, the ratio between the concentration of the virus and the number of cells of a particular type. C, the pH temperature environment around different cell types. Or D, the availability of virus products, secreted by different cell types. So I'm gonna read that again. The availability to viruses of products secreted by different cell types. So let's do a brief overview real quick of how a virus infects a cell. And I'm going to pull up a picture here so we can see. So basically we have a virus and we have a cell. And in order for the virus to connect to the cell, it has to have a matching lock and key sort of thing, if you will. The cell has the lock, the virus has the key, they connect, and the cell opens and the virus comes in. So now let's go, I'm going to leave this picture up and we're going to go back and look at each of these answers again. So again, we're going to start with the incorrect answers. So we're going to skip A because in this case, this is the correct answer. So let's start with B. So B is incorrect. The ratio of virus and cell types are not going to tell us which cells are infected. C is incorrect because the pH and temperature of the body are constant and are not going to differ around healthy versus unhealthy cells. So it's not going to make some cells more likely to be infected than other cells because it's the same pH and temperature. D is incorrect because cells do not become infected by what they secrete. That will not make a cell more likely to be infected. Again, it's whether that lock and key match sort of thing. So now let's go back and look at A. So A is correct because the ligand on the surface of the virus, which is like the key, and the receptor on the surface of the cell is like the lock. And that's how the virus is able to get in when the correct ligand and find the correct receptor on the cell. So in this case, A is going to be correct because that's the only one that matches here. So let's go back now and look at question 8. So 8 says, which of the following is a property of carbon that makes this element basic to the chemistry of life? A, carbon has many isotopes, 2 of which are stable. B, carbon atoms are arranged in different ways in different crystalline structures. C, a carbon atom has 4 valence electrons and forms 4 covalent bonds with other atoms. D, a carbon atom has an equal number of protons and electrons. So again, let's start with the incorrect. So A is incorrect. So there's a lot of vocabulary kind of going on here. So the first thing, let's define what an isotope is. So an isotope is an element with the same number of protons, but a differing number of neutrons. So they share the same properties, but their masses are going to be different due to the differing number of neutrons. Another word they talk about here is stable. So stable means it can hold the same, it can hold on to that and it'll stay the same. Unstable means it's going to experience nuclear decay. So these are going to exist in all elements and are not specific to carbon. So same with the idea that of some being stable and some being not stable, all of that is common to lots of different types of elements. So that's not going to explain here why carbon is the basic chemistry of life. All right, so let's go on to B. B is going to be incorrect because carbon's ability to form different kinds. B is incorrect because carbon's ability to form different kinds of crystalline structures is not going to influence carbon's role in the chemistry of life. So its ability to form different structures doesn't make a difference to why it's more important. And then D is incorrect. While it's a true statement, most elements have the same number of protons and electrons. So this is not unique to carbon, therefore it doesn't make carbon any more valuable than anything else. So in this case, C is going to be the correct answer because the number of valence electrons, the number of valence electrons is determines how many covalent bonds it can make. So being that it can make 4 bonds, that's more than a lot of atoms can make. So if you need more review on valence electrons, I have a video here. Let's see. Oh, the link got deleted. Hold on, let me find the link. Let me put the link here. Back so you guys can see it. See if that's gonna, there it is. Okay. So you can see here. There we go. So here's a link basically that will give you more review over valence electrons and bonding if you're needing some more review on that. But basically, carbon has 4 valence electrons. It can therefore create 4 bonds, 4 covalent bonds, which makes it more valuable in chemistry because it can create more bonds. All right, question 9 here. We've got a diagram, so we need to make sure we pay attention to our picture. So a model of a eukaryotic cell membrane is shown above. Which of the following is the most likely function of the structure labeled X? So we're wanting to look here at what is the structure of this right here. So A, facilitating the exchange of O2 or oxygen and CO2, carbon dioxide, by the cell. B, catalyzing the synthesis of molecules secreted by the cell. C, promoting the entry of hormones into the cell. Or D, enabling the movement of ions across the cell membrane. So again, let's start with the incorrect answers. So A is incorrect because oxygen and carbon dioxide can basically go directly through the cell membrane. They don't require assistance of a membrane protein, which is what X is. This right here is a membrane protein. So carbon dioxide and oxygen are both very small. They're not charged. They can go straight through the membrane without a problem. They don't need a membrane protein. B is incorrect because nothing is being made in X. It's a protein in the membrane, as we said, used to transport things. It doesn't make anything. So the question is, what's being transported? Something being made in the cell, that's going to be done in an organelle deeper in the cell. That's not going to be on the membrane. C is incorrect here. There are two types of hormone proteins. So C, it says hormone proteins entering the cell. So there's two types of hormone proteins. One is going to be a protein and one is going to be a lipid. Protein hormones don't typically enter the cell. They just bind to the outside. So they don't need to be able to go in and out. So that eliminates that part of a hormone. The other types of hormones we have are lipid hormones, and those can enter the cell, but they can go directly through the cell membrane since the cell membrane is made of lipids. Like can go through like. So a lipid hormone could go straight through, it wouldn't need a protein. So our correct answer here is going to be D. So D says enabling the movement of ions across the cell membrane. So these are going to be big charged molecules that can't go directly through the cell membrane. They have to have a protein to help it go across. So they need a channel. So since X is a channel protein, that's going to allow those molecules to go through. The only things that can go directly across are going to be small molecules, which is why A doesn't work. Those O2 and CO2 can go straight through the membrane. Or lipids, such as a lipid hormone that could go straight through the cell membrane. So small things can go straight across, or lipids because the membrane is made of lipids. So just, I want to briefly go over if you're needing review on, oops, here we go. The different types of transport. This is just kind of a short overview of transport, but basically we have active transport and we have passive transport. So passive transport doesn't require energy. Things can go through the membrane or the channel protein in this picture without needing energy. And the way they do that is they're going to go from high concentration to low concentration. Just like, for example, if you were to take a bottle of, let's say, perfume, and you were to spray it, you only sprayed in a small section of the room, eventually you're going to be able to smell it throughout the room. The idea is that those perfume molecules are going to go from that high concentration and they're going to spread out throughout the room, filling in the lower concentration. So all of these pictures under passive transport, you can see there's higher concentration outside, lower concentration inside. So that's going to drive the movement of these molecules. So there are three different types of passive transport. There's simple diffusion. You can see here, they're going straight through the cell membrane. That would be either small molecules or your lipids, as we just talked about. Those things can go straight through. The other two types the other type we have is facilitated diffusion. So that's, you can see here, that is going to be what requires a channel protein, which is what they're talking about in our question. So we're still going from a higher concentration to a lower concentration, but these can be bigger molecules or non-lipids that need extra help getting across to follow that concentration. And then the last type of passive transport is osmosis, and that's only water. Water going from a high concentration to a low concentration. And then the other type of transport, so those are all types of passive transport. The other type is active transport. And active transport, as you can see from the picture, is going to require energy. And as you see here, these are molecules that are going from lower concentration to higher concentration. So in this picture, you can see we have 2 diamonds here. We've got 1, 2, 3, 4, 5 here. So they're going from low concentration to high concentration. We call that a concentration gradient. So they're going against their concentration gradient, which is why they need energy here. So again, this is just a good, simple little overview of the different types of transports. All right, so last question in this section. Which of the following properties best distinguishes prokaryotic and eukaryotic cells from viruses? So things that are common to prokaryotes and eukaryotes, but not to viruses. So A, prokaryotes and eukaryotes divide by mitosis, whereas viruses do not. B, prokaryotes and eukaryotes synthesize proteins on ribosomes, whereas viruses do not. So C, prokaryotes and eukaryotes are surrounded by a lipid bilayer, whereas viruses are not. And D, prokaryotes and eukaryotes have genomes of DNA, whereas viruses have genomes of RNA. So again, this is somewhat of a two-part question. We have to know the similarities of prokaryotes and eukaryotes, what makes those similar, and then what makes those two different from a virus. So before we get into the correct and incorrect answers, I want to go over some of the major differences between prokaryotes, eukaryotes, and viruses. I'm going to pull up a chart here so we can look. So this chart is not going to have all of the differences, but it's going to have some of the biggest ones. So let's quickly go through look through this chart and then see if we can use this chart to at least eliminate some of the answers here. So prokaryotes are going to be bacteria. Eukaryotes are plants and animals. Viruses here, it says those can contain DNA or RNA. Prokaryotes and eukaryotes both contain ribosomes. Viruses don't have a nucleus, so they do not contain ribosomes. Prokaryotes and eukaryotes both have DNA and RNA. I don't know why they have these switched here. Versus viruses have a capsid surrounding that genetic material, and that can be DNA or it can be RNA. You have DNA viruses, you can have RNA viruses. Prokaryotes have no nucleus versus eukaryotes do have a nucleus. And no membrane-bound organelles, which again, here, viruses don't have membrane-bound organelles. You can see down here over in prokaryotes, they also do not have membrane-bound organelles. Not sure why they didn't put those next to each other in this chart. Prokaryotes are going to be unicellular organisms, one cell, versus eukaryotes are going to be multicellular, right here. Viruses are too small to be seen on a microscope. Too small to be seen even with a light microscope. So viruses, like when you're in the science lab and you can look at an animal cell or a plant cell under the microscope, viruses are so much smaller than that you need way more. And viruses are not a cell. So prokaryotes and eukaryotes are both cell types. I'm sorry. Viruses are not a cell type. So I'm going to leave this chart up there while question are we on here? Question 10, correct? Yeah, well, we go back through the answer choices. So A, prokaryotes and eukaryotes divide by mitosis, whereas viruses do not. This answer isn't actually found on this specific chart. As I said, not all of them are on here, but only eukaryotes divide by mitosis. Prokaryotes actually divide by a process called binary fission, and viruses don't actually divide at all. So A is not correct. If we look at B, so using our chart, we can see that B actually is correct. Viruses do not contain ribosomes and therefore cannot synthesize proteins on ribosomes. This is why they infect our cells or bacteria cells. They need our ribosomes to create those proteins. So they infect our cells, use our ribosomes to create those viral proteins going forward because they do not have the ribosomes to be able to do that. C is incorrect because viruses on their own do not have a lipid bilayer. Some can get some once they infect an animal cell. Once they come into our cell, then they leave Sometimes they take some of our lipid bilayer with them, but viruses on their own do not have a lipid bilayer. And then D is incorrect, as you can see from the chart. Viruses may have RNA or DNA. They can have either one. So that should be our last question from this, from this exam. Yes, so for this is all for this first section for questions 6 through 10.